Hey, seventh graders, we are winding down on our DNA unit, and the last lesson for this unit is going to be a little talk on mutations. And mutations is something that we're going to talk about for the rest of the school year. So keep that in mind that it's going to be a big topic or focus for us. So the biggest thing to remember is that mutations aren't necessarily good, they're not necessarily bad. More often than not, we don't see anything happen with mutations. But in the case of today's assignment, you guys are going to kind of research why a mutation is either good or bad, or what does that do to cause changes. So if you guys remember, we talked about DNA being the blueprint for life, and that it gives us the instructions to make proteins. And so if we change the instructions, essentially we could be making a different protein, or we could be making something that is exactly the same as it was before mutation or not. And if we're making a different protein, it could be something that's good or bad. Okay, so your learning target for today, I can describe a mutation in DNA as either good or bad and explain why. And I can explain how a mutation might change the structure of DNA or protein. So that's really what we're going to get in with this simulation today. For your bell ringer today, do you think mutations are good or bad? Circle either good or bad, and then explain why you chose that. Okay, if you guys need to take a second and pause and complete your bell ringer, go ahead and do so now, and then we're going to move on to the first part of this assignment. Okay, so for this, you guys are going to need to open this mutations link. It changed a little bit in the past couple years, so I had to redo this video. Your link now looks like this. And what's different about this now is we can input our own DNA sequence. So what you guys are going to do is you're going to highlight, you're going to hit Control-C, and you're going to go grab that DNA sequence. It used to just give you a set DNA sequence, which we could then mutate, but in this case, we can kind of add our own DNA sequence. So click resume to see your DNA turned into protein. As you know, DNA gives us the code that makes proteins, and the proteins help build our body and make it function. We're going to look at how mutations in our DNA change the proteins we build in our cells and bodies. So you guys are going to go to edit or add in a DNA sequence, and we're going to paste Control-V, our copy there. Okay, and we're going to hit apply, and then we're going to go down here to resume, and we're going to watch what happens with that DNA. Okay, so we go to the nucleus of our cell. We find all of those little squigglies there are our DNA, and we can zoom in on our little double helix right there. And it's going to show us the strand that we just inputted. Okay, and then it's going to take this little, shoot, where am I at? Sorry, it slowed down on me and stopped. Okay, so it's going to go ahead, it's going to split our DNA into two strands, and then we're going to make a copy of what we call our mRNA, which is messenger RNA. That messenger RNA has the ability to leave the nucleus, whereas our DNA doesn't have the ability to leave a nucleus. So we're going to go ahead and turn that DNA into mRNA, and that messenger RNA is going to leave our nucleus. It's going to go outside, and we're going to find what we call a ribosome. And that ribosome is going to be our little chef or our little builder, and it's going to take and read the instructions in that messenger RNA, and it's going to turn it into a protein. So here we see our amino acids and our protein being built. Remember, amino acids are the building block. So each one of these little circles is another amino acid. We have 20 different types of amino acids. So this is going to be our regular unmutated strand of DNA is going to form this protein. The biggest thing that you guys need to remember is that proteins are kind of like a key, and if we change the shape of the protein, that key's not going to function in the same lock anymore. So if we don't change the shape, that key's probably fine, but if we change the shape, it's not going to be fine. So then what you guys are going to do is you're going to screenshot. I'm going to use my snipping tool on mine, and you guys are going to take a screenshot of what that protein looks like. So if I go ahead and click mine here, it's going to kind of pause for me, and then I'm able to screenshot it, copy it, and paste it. Okay, so if I look on the next page, I've already done the normal strand for you guys. You guys will still need to input that DNA and figure out what you need to do for your mutations, but here's your normal strand. One thing I've also done, which is kind of a nice feature with this, is I clicked unfold protein, and it kind of straightens out my protein for me. It's not wanting to go here. Okay, refold, unfold. Okay, for whatever reason, this is not wanting to line out for me. So what I've done is you guys will see a copy of what it looks like unwound. Okay, so you guys have 7 mutations you guys are going to try, and you're going to use that unwound protein that I put on the next slides for you guys to kind of compare how your guys' mutated proteins differ. So now try 7 mutations to see what effects those mutations have on the proteins they are supposed to make in comparison with the normal protein. Generally, if a protein is different, it will also function differently. Think of a key. If you change the shape of a key, it will no longer work in the same lock. For this activity, you will click Enter or Edit a DNA sequence and change the 8th base pair of your DNA strand. So that's the 8th letter. In the normal DNA, it is a G. Try one of each mutation for that 8th base pair and resume to show your protein. Screenshot your proteins, compare to the normal protein. Highlight where the protein is different if there is a difference. Click enter or edit a DNA sequence to make your next mutation. So what this is going to look like is I've already kind of done this for you guys. What you're going to do is we're going to go in and we're going to change that 8th G to an A. So I'm going to go to edit a DNA sequence and I'm going to count 1, 2, 3, 4, 5, 6, 7, 8. So here is my G and I'm going to change that to an A. So I've just made a mutation and then I'm going to hit resume and we're going to see how that protein is now different. Okay, so it's going to go ahead and build my little protein chain for me of amino acids. And then what we're going to be able to do is compare those little shapes of each of those amino acids to say, all right, are any of these amino acids different? Okay, at the end of this, you guys should see that it kind of has, if there's a big mutation, it might have kind of a different shape. So it's on the unfolded version right now. So we can kind of refold it and see what it looks like. Okay. So that's kind of a little bit different shape than what we normally have. If I hit unfold protein, you guys can see it all laid out in sequence. So if we look here on my 2 screenshots, here's it all folded up, and then here's it all straightened out. Down here is a picture of my normal version. So if I take and I look at this, I'm going to grab a draw tool really quick, and I'm going to say, all right, it starts with this triangle. Here's my triangle. Then we go to this oval, and then here's a difference right here in this protein right here. Instead of this sideways oval, we've got this little hexagon. Okay, and then we go circle, diamond, hexagon again, diamond, half circle, Cheerio or whatever you want to call it, square, and then again another half circle. Okay, so if we look at that, this is the only difference that we see is this one here. So what I would do is take my highlighter and circle that. Okay, or if I want to do an unfolded protein, that's totally fine. But what you guys got to remember, it's really the shape that's important on this. So if we look at this, this is going to have a different shape than what our original one had up here. Okay, but because of that one difference in this amino acid right here, that's why we have a different shape. So however you guys choose to do it is fine, but then you guys would take your highlighter, whether you do the folded up or if you do the unfolded, you're going to highlight the one that's different. So I would highlight this little stop sign one that wasn't in there before. So this could be something that makes you more adapted to your environment, makes a better protein than what you had before, or it could be something that is not good. It could maybe lead to a disease or oopsies, or you know, whatever the case is, it makes you less suited for your environment. So keep that in mind that we could see something good or bad. You guys might see no change at all. Even though you guys mutated your DNA, you might see that you get the same sequence of shapes and that you have no change. If that's the case, I want you guys just to write on there with their draw tool, no change. Okay, obviously we saw a change, so this wouldn't apply to this problem, but you guys would just write on there no change. Okay, meaning that your protein was exactly the same, and even though there was a mutation, it didn't cause any change in your DNA. So we wouldn't even see any effects of that mutation, whether good or bad. Okay, for this next one, you guys are going to substitute G to T. So G to A was already done for you. So again, this one will go to edit or edit, whatever, and we're going to change this to a T instead. Okay, apply and then hit resume, and it's going to rebuild that protein. So then you guys will screenshot there. You guys will compare it to your normal sequence down here, and you guys will use your draw tool to highlight any differences. Okay, for your last one, you guys are going to go a C to a G. So now change that, or excuse me, a G to a C. So now you guys are going to change your sequence. So this one's going to be now a C instead of a T. Okay, and then for your next one, you're going to do what we call an insertion. So we're going to add an A. So right in front of where that G normally was, so this was a G, so you guys can go back to that original G. There we go. You guys are going to add an A right in front of that G. Really doesn't matter where you guys add it. Before or after, you guys will see a difference. Hit apply and resume. Okay, and then this one we're going to do add a T. So we're going to insert and add a T, add a G, add a C. And then you're going to remove that. In this case, we're going to remove the G entirely. So for the last one, you guys are going to just straight up get rid of this letter. Okay, so now there's one less letter. Okay, there goes our G. Okay, so you guys will follow those instructions. Each time if you need to just kind of reset to do your mutation, that's totally fine, especially when you guys get to the point where you're doing your insertions. Feel free to go back to slide 3 and copy and paste the original DNA strand in there. So that way, when you guys are doing the mutations, you guys make sure you're doing the correct mutations and not like over mutating one strand of DNA. So if you guys are unsure, like I said, when you're done with your substitutions, just go and copy and paste your old DNA or your original DNA, and then that'll be a lot easier when you guys add these in. Okay, for the next part of this, it's called Guess the Mutation. So you guys are going to have a couple different mutations for you. So on the top, we have a normal DNA. On the bottom, we have a mutated DNA. So you guys are going to compare the two and say, all right, the C and G, AT, here we've got something different. Okay, so if I look, this one is lined up with this one. Okay, and then all of these from here on out are all lined up. So you guys can kind of compare the two, draw if you need to. Okay, but what I see with this is we have one extra base pair right here. So this would be what we call an insertion because a deletion, because in the normal DNA, we have one extra base pair, and then in the mutated DNA, we got rid of it. So you guys would highlight deletion, and then you guys would say, all right, where did we delete that? This would be the 1, 2, 3, third base pair. So I would circle third base pair on that, and then describe the change. The AT was deleted. So I kind of already did that one for you guys. So you guys have kind of just finished that. So this is kind of what it should look like filled out. It was a deletion. It happened on the third base pair, 1, 2, 3. The third one got deleted. And then you'd write here, describe the change, the AT was deleted. Okay. Then you guys are going to look at the other. So this one, again, compare the two. On the last one, you guys have 2. So this is extra credit challenge. If you get this correct, you get an extra credit point. Mutations. What you're going to do is you're going to use 2 different colors. And so you're going to highlight, like maybe if I do on the first mutation, I want to do it in green. Then my second mutation, I'm going to do it in pink. So I would still circle either, my green one, what kind is it? I'd circle something in green. My pink one, which is my second mutation, what am I going to circle for that one? So I should have 2 colors up there. And then what number did my green one, my number 1 happen on? What number did my number 2 happen on? And then I'm going to describe both of those changes. And then you guys can kind of just highlight those green or pink depending on where they were at. So that's your guys' extra credit for that. Okay. Other than that, you guys are good. Great job, you little mutants. Now hand it in. If you guys have any questions, let me know, and I hope you're having a great day.